Welcome to the Deep Dive. We explore compelling research and break it down so you're instantly informed. Today, we're diving into a single, really fascinating paper. It might just change how we monitor plant life, basically by sampling the air.
Yeah, the paper's called Airborne Environmental DNA Metabarcoding Detects More Diversity with Less Sampling Effort Than a Traditional Plant Community Survey. Came out in 2021, lead author Mark D. Johnson.
And this is pretty big news in the field. It takes this emerging tech airborne eDNA, environmental DNA, and uses it in a totally new way. I mean, people have used airborne eDNA before, but usually for specific things, right? Like tracking pollen counts or maybe airborne pathogens.
Okay.
What this team did, Johnson and colleagues, was they used this advanced genetic sequencing called metabarcoding to look at everything. The whole plant community's DNA signature floating in the air. And then, crucially, they compared it directly to the old school methods. Okay, let's unpack this then, because, we have botanists, experts who are really good at identifying plants on the ground. Why exactly do we need, like, a fancy air filter to tell us what's growing nearby?
That's a fair question.
Yeah.
It comes down to needing accurate data. Knowing exactly what plants are where is just fundamental for conservation, for managing land properly, for restoration projects, all that stuff. But the traditional ways of doing it, they have some serious drawbacks. Think about the standard visual survey, maybe using transects. First off, it takes a ton of resources, people's time, money, expertise.
Right.
And second, it's really dependent on the skills of the person doing the survey. You get observer errors. Different people might identify things differently, or even the same person might make mistakes sometimes. It's subjective.
And that method you mentioned, line point intercept, what does that actually involve? Sounds kind of painstaking.
Oh, it is. Often it's literally stretching out a tape measure, walking along it, and at set points, dropping a pin or pointing straight down and recording exactly what plant species that point touches. Yeah, it's systematic, but incredibly slow. And think about it, you're trampling through the area, potentially disturbing the plants, maybe even local animals. It's physically disruptive.
So the promise of eDNA here is, well, it's faster. You don't necessarily need a top botanist just to collect the sample, and it's less disruptive. We've seen this work really well in water systems, haven't we?
Absolutely. There are some great examples, like detecting invasive silver carp DNA in Chicago waterways before anyone could physically catch them, or finding invasive bullfrogs in France just from water samples. eDNA often picks things up. Traditional methods miss in aquatic environments.
So the huge question this paper tackled was, can we get that same kind of sensitivity, that power from the air, which is obviously much more dynamic than water?
Exactly. And to test it, they set up this really thorough comparison study.
Okay, set the scene for us. Where did they do this work and when?
They ran it for a full year, June 2018 to June 2019. The location was a short grass rangeland, basically native prairie, managed by Texas Tech University. They picked out 9 specific sites within that rangeland for the comparison.
9 sites. Okay, and they compared the two methods there.
Right. So method 1, the traditional survey, they did 2 big visual surveys, these intensive efforts. One in September 2018, so capturing the fall plants, and another in May 2019 for the spring bloom. Using those line point intercept methods and general visual identification, trying to get a best possible snapshot with the standard approach.
2 snapshots in the year. And the eDNA side, what were they using to collect the air samples? Some kind of vacuum?
Actually something simpler called Big Spring Number 8 dust traps, or BSNE traps. It sounds a bit low-tech maybe, but they're passive collectors, basically designed to catch airborne dust and particles, which includes the eDNA.
Okay, passive traps. And how often did they collect from those?
This is key. Because they were simple to manage, the team went out roughly every 2 weeks for the entire year to collect the samples from the traps at those 9 sites. That gave them 22 collection events in total.
22 collection points versus just 2 big survey events. You can already see a massive difference in the potential data right there. But how do you get from, well, a filter full of dust to a list of plant species?
That's the metabarcoding part. It's really powerful. Instead of looking for the DNA of one specific plant, like you might with older methods, metabarcoding sequences a whole bunch of DNA snippets all at once.
Like a genetic census of the sample?
Kind of, yeah. They targeted a specific gene region called ITS2. It works like a pretty good universal barcode for many plants. So they sequence all the ITS2 fragments in the dust sample, and then they match those sequences against massive online databases of known plant DNA.
So match the barcode, identify the plant.
Basically, yes. It lets them see the whole mix of plant DNA present in that air sample. It's looking at the total biodiversity signature captured in the dust.
Okay, so they did all this work, a year of sampling, complex lab analysis. What was the big headline finding when they compared the airborne eDNA results to those two traditional surveys?
Well, the first major point was about diversity. The airborne eDNA method detected more species overall. It found 91 unique species across the year.
91. And the traditional method?
The traditional surveys found 80 unique species that had reference sequences available for comparison. We need to come back to that caveat, it's important. But based on comparable data, eDNA found more.
Okay, more species detected. But you mentioned efficiency earlier. That sounds like the real game changer here. Tell me about the effort involved again.
Right, this is where it gets really striking. Each of those traditional surveys, the September one and the May one, took literally hundreds of volunteer hours. Plus, you needed those expert botanists on site for the identifications.
Hundreds of hours for 2 points in time. Okay. And the eDNA work. Collecting and processing those filters from 9 sites 22 times over the year.
The paper estimates it took about 66 hours total for one person.
66 hours for a whole year of continuous monitoring versus hundreds for 2 snapshots.
Exactly. The difference in sampling effort is just enormous. You're getting potentially richer data, definitely more frequent data, covering the whole year for a tiny fraction of the labor.
And that frequency, that temporal coverage must be huge. It means you can catch things happening between those big survey events.
Precisely. They saw this really clearly with a plant called tansy mustard. The eDNA data showed huge spikes of it early in the spring. It was obviously having a massive bloom event.
Okay.
But by the time the traditional survey team went out in May, that bloom was mostly over. The plant wasn't very common visually anymore. So without the frequent DNA sampling, that whole ecological event, that big pulse of tansy mustard, would have been largely missed by the traditional snapshot.
That makes sense. And that ability to sample frequently and easily must be critical for catching invasive species early, right? Before they become a huge problem.
Absolutely crucial. And they found a perfect example. The eDNA surveys detected tree of heaven Ailanthus altissima, which is a notoriously bad invasive species. It picked it up when it was still rare on the site, not really established yet.
In the traditional survey?
I missed it completely. Finding invasives early, when you might actually have a chance to eradicate them cheaply, is the holy grail. Airborne eDNA seems like a really promising tool for that kind of early warning.
So eDNA found more species overall in the comparison. It was way more efficient, and it caught things like rapid blooms and early invasives. But did it just find different things? Or was it better across the board?
That's another fascinating part. The study showed the two methods actually complement each other quite well. They have different strengths, or maybe different biases, you could say.
Okay, biases based on what? How the plants live.
Pretty much. For example, the airborne eDNA was much better at detecting grasses. It found 13 unique grass species that the visual surveys missed entirely. The visual surveys only found 2 unique grass species that eDNA missed.
Why such a big difference for grasses?
2 likely reasons. 1, grasses are typically wind-pollinated. They literally throw huge amounts of pollen, their genetic material, into the air, so they create a strong airborne signal. 2, grasses can be really tricky to identify visually, especially if they're not flowering. eDNA bypasses that identification challenge.
Okay, so eDNA is great for wind-pollinated things like grasses. What were the visual surveyors better at spotting?
They tended to pick up rarer plants that had big, obvious, showy flowers, things that naturally catch your eye when you're walking a transect. The eDNA, well, it doesn't have eyes. It's just passively collecting what's in the air, so it was better at detecting plants with maybe smaller flowers or different kinds of pollination strategies that don't rely on being visually obvious.
So the plant's lifestyle really determines how easily each method detects it. Can you give us a couple more examples of that?
Sure. Take blue grama grass. This was actually the most common species found by the visual surveyors. But the eDNA told a different story over time. Its signal was really high in the eDNA samples during the fall.
When it was pollinating.
Exactly. But then in the spring samples, when the grass was dormant and not releasing pollen, its eDNA signal dropped way down. The air signal tracked its biological activity.
Interesting. And what about something with a completely different strategy, like Russian thistle, the tumbleweed?
Oh yeah, Russian thistle, Salsola tragus, a perfect example. It was consistently one of the most common species found in the eDNA samples, right through the year.
Why is that?
Because its whole strategy involves the mature plant breaking off at the base and tumbling across the landscape, spreading seeds and plant fragments full of DNA everywhere it goes.
So it's constantly shedding material into the environment.
Constantly. That makes it hard for a stationary visual survey to get a consistent measure, maybe, but it guarantees a strong, persistent signal in the air traps.
the study didn't just look at what was detected, but also where, right? They mentioned wind patterns.
Yes, they did.
Yeah.
And it confirmed what you'd intuitively expect. The main wind direction at their Texas site was from the northwest. And guess what? The traps located upwind, in the northwest, consistently collected the fewest species. The traps in the center of the study area, and especially those downwind in the southeast, collected the most species.
So the wind is literally acting like a conveyor belt, carrying the DNA signatures across the landscape.
It really seems that way. It accumulates the signal as it moves across the plant communities, which also suggests the signal isn't just super localized pollen, right? It confirms that we're picking up fragments of leaves, flowers, maybe even just free DNA floating around.
Which has huge implications. It means this technique could potentially track changes in whole communities over time due to, say, climate change or disturbances. But what are the catches? What's stopping everyone from just deploying these air traps everywhere tomorrow?
Okay, yeah, we need to talk limitations. And there's a really important one we hinted at earlier, the reference database gap.
Right, you said the traditional method found 80 species that could be compared. What about the ones that couldn't?
This is the crucial point. The visual surveyors identified 22 plant species on the site that they couldn't compare to the eDNA results. Why? Because those 22 species didn't have their IDES2 barcode sequence lodged in the big public databases like GenBank.
Wait a minute, 22 species missing from the comparison. If you added those back into the traditional survey total, that would be 102 species, wouldn't it? Which is more than the 91 eDNA found.
Exactly, and that completely changes the eDNA finds more diversity headline, doesn't it? It shows that the eDNA technique is fundamentally limited by the completeness of our genetic reference libraries. If a species' DNA isn't in the library, metabarcoding can't identify it, even if it's present in the sample.
So the technology's power is directly tied to the background data we have. That seems like a major bottleneck right now.
It is. It's probably the biggest bottleneck for widespread eDNA use in general, not just airborne. As more species get sequenced, this problem will shrink. But right now, it means eDNA alone might miss things we know are there visually, simply because we lack the reference barcode.
That's critical context.
Yeah.
Did the study reveal anything else about the best way to actually do the sampling or analysis?
Yes, one practical finding. They tested pooling samples, basically mixing the dust from all 9 traps together for each collection event before sequencing, probably to save money.
Did that work?
It worked, but it wasn't ideal. They found that analyzing the traps individually detected significantly more species than analyzing the pooled sample. Something about the pooling process seems to mask some of the rarer DNA signals.
So if you can afford it, analyze each trap separately.
That seems to be the takeaway for maximizing the diversity you detect, yeah.
Okay, so summing this all up, where does this leave us in terms of monitoring plant life? What's the big picture conclusion?
I think the core takeaway is that airborne eDNA metabarcoding is an incredibly powerful tool. It's highly sensitive, especially for certain types of plants like grasses. It's vastly more efficient in terms of effort, and its frequency gives you temporal resolution you just can't get easily with traditional methods. Think about tracking those bloom events or invasive species introductions.
But it's not perfect, mainly because of those reference database gaps.
Right, which is why the paper concludes, and I think most researchers would agree that the best approach, at least for now, is probably a combination. Use the strengths of both. Traditional surveys are still essential for finding those visually distinct rare species, for ground truthing, and for dealing with species not yet in the databases.
And use airborne eDNA for the broad-scale, high-frequency monitoring, the efficiency, tracking changes over time, and potentially catching those hard-to-spot or wind-dispersed species.
Exactly, they complement each other.
So what does this all mean? It really feels like the air around us is this constant flowing census report of the plant life nearby.
That's a great way to put it, an invisible stream of information.
And it leads to a final kind of provocative thought based on something else they found. You mentioned the databases when they checked their sequences. They didn't just find local rangeland plants, did they?
No. Using a detailed analysis tool called BLASTERN, they found clear DNA matches for things like cotton and soybean.
Which weren't growing on their native rangeland site.
No, but they were likely growing in agricultural fields maybe up to a mile away. The wind carried those signals too.
So think about that. If airborne eDNA is traveling potentially miles on the wind, how could we maybe refine this to track the spread of an invasive species across a whole region? Or, flipping that around, what DNA from distant farms or forests or even cities are we all breathing in every single day without realizing it? It suggests there's this huge invisible information network in the air that we're only just starting to learn how to read. What stories are floating past us right now?
That's the next frontier, isn't it? Decoding that aerial library.